Hello to all of you. Let's discuss chapter number 8, and we are going to start discussing from where we left. So let me go to the slideshow mode. So basically, we have been talking in our lecture. We talked about how the information which is there in genes is converted into proteins. So we have discussed the structure of protein. We have discussed it's made of amino acids, and we also discussed the gene expression, that it has two steps: transcription and translation, that the information which is there in DNA, first of all, it's converted into mRNA, and then that mRNA goes to the cytoplasm. There it binds on the ribosomes, and then the ribosomes move on the mRNA, and then the information in the RNA converted into polypeptides. So basically, we have covered up to here, and this is the slide which we have discussed in our chapter number 7 also. So in this slide, we basically talked about how the changes which can take place in the genes, in the DNA, and can lead to the formation of abnormal proteins, and if those proteins, they are involved in cell cycle, then they can lead to the aberrant growth of the cell. So this is basically the same slide. So because now right now we are talking about DNA to protein, so it basically makes more sense now that all these changes like gene amplification, point mutations, chromosomal translocations, and various other events, these takes place in the DNA, they take place in the genes, and they cause the changes to take place in the proteins. And here is the example of some of the proteins which get overexpressed in cancer. Example is the membrane proteins like EGFR and HER2. These are two membrane proteins which get overexpressed in cancer. And there are more examples of some enzymes like hexokinase, phosphofructokinase, and pyruvate kinase. So these are some of the proteins, basically the enzymes, which get overexpressed in cancer. And this is the example of some of the proteins which get mutated in cancer, which means they get changed. The example is PI3 kinase, AKT, MAP kinases. These are all proteins which are involved in cell proliferation and cell survival. And there are mutations which also takes place in some of the growth factor receptors like EGFR and HER2, and mutations also take place in some of the tumor suppressor genes like P53 and P10. And now the last part of this chapter is about like how we can make the proteins um, in the lab basically. So basically, this is a field of biotechnology called gene cloning, or it's called recombinant DNA technology. So in this technology, making use of this technology, what is done is that you can take the genes from different organisms, and you can express them in the bacteria. Now, for example, let me give you an example like insulin. Insulin is a medicine basically which is used for diabetic patients, and insulin is nothing but a protein, and it is coded by some genes. Like if a human being, like if a human person is, if there's a patient who is suffering from this disease and diabetes, then this person needs to be given insulin. Insulin is nothing but a protein, so it has to be, earlier it used to be actually this insulin used to be extracted from some animals, and it used to be purified and given to the patient, so it's quite expensive process. But now with the help of the recombinant DNA technology or gene cloning, we can take the genes and we can clone them. So we can take a human gene which codes for insulin, and we can clone it, which means we can make multiple copies of that gene. We can make multiple copies of that gene, and that gene is useful. That insulin gene is useful if that gene can be coded into insulin protein, because the medicine is actually protein. So now we are going to talk about this technique called recombinant DNA technology or gene cloning, by which we can make multiple copies of the proteins, and we can bring the cost of those proteins, which are basically used as some drugs, we can bring their cost down. So this is the technology which is being used these days to make various drugs. And here is example of the spider silk. Now spider silk is basically the spiders, they have a gene for the silk, and we can, so it's a naturally present gene in the spiders. So we can take this gene and we can clone this gene, and we can produce the protein which is coded by the silk gene, and we can have it means by cloning the silk gene, and if that gene we can make it code or express into proteins, we will have a lot of silk protein. And why we are interested in silk? Because of various advantages of the silk. Spider silk is tough, flexible, elastic, making it well suited for military and industrial use. So you can see in the military, dress and the vests and all that, we can use this spider silk. It is biocompatible, make it ideal for variety of surgical applications. So basically, like these are some biocompatible and biodegradable fluids and sutures. We can make use of the spider silk and make all these things with the spider silk because it's biocompatible. And these are few more applications of the silk, like it can be used as a textile, skin grafts and all. So it means that we are interested in the silk. So how we can clone the silk protein? It's a basically, whether it's a insulin, it's silk, or any growth factor, the technology remains the same. We take the gene out from the host, and we basically where the gene actually is present, like for the spider silk, it is present in the spiders. So we can take the gene out, and we can fuse this gene with the gene or a regulatory gene of the host. Like we can put this gene either in bacteria, or we can put this gene in the yeast, because these are very fast growing organisms. So in this organisms, the gene actually multiplies quite fast, and it means that when the gene multiplies or amplifies, it codes for the multiple copies of the proteins. So we can take basically, it's making a kind of a fusion gene. We can take the spider gene and fuse it with the yeast regulatory gene and form a recombinant gene, and so we form a recombinant gene, and then this recombinant gene is actually transferred to some vehicle, or we call it a vector. DNA vectors, they're usually plasmids. We can put this gene in them, and then we can use these vectors to make this recombinant gene to go into the yeast. In the yeast, cells grow very fast. In this picture you can see they're showing some reactors. So we can grow the yeast cells here, and from the yeast cells we can actually yield, or we can have this protein. So this is a technology, this is called gene cloning technology. So making genetically engineered silk begins with isolating spider DNA from spider cells. You can see here, this is a spider, this is a gene, we can take it out. This DNA can be inserted into a cell of a new organism. So the like the yeast, when we put this spider gene into the yeast, this is organisms. So organisms that have been genetically modified to contain genes from other species are called transgenic organisms. So for making a recombinant gene, as I said, we are coding gene from the spider. If we are cloning the spider gene, we take the gene from the spiders and we fuse it with the regulatory sequence from the host, which is yeast here. And what is the regulatory sequence? It's a on and off switch of the gene, and we can then put it into the we make a recombinant gene, and then we put it into a vector, and then that vector we put it into the yeast cells. So this is a recombinant DNA technology, and this technique is used for the treatment of cancer. So there are some drugs which are given to cancer patients, and they are actually made with the help of this gene cloning technique, which includes tumor necrosis factor and the interleukin 2. These are both two drugs which are basically proteins which are basically used for the treatment of cancer, and they are produced by the gene cloning technique. So I hope you understand the chapter. If you can let me know, you can send me email, or I can also meet you virtually through Blackboard Collaborate. But if you want to meet me, have a virtual meeting, you have to tell me at least 1 or 2 days before, so that I can set up a meeting for you guys there.